Hello, in this video we are going to continue talking about the adaptive immune response, specifically in how it relates to B cells and their ability to produce antibodies. First, just a little bit of history about B cells. So B cells were the first uh, component of the adaptive immune response that were characterized mainly because, or when I say they were, I mean the fact that you had antibodies were. And since B cells produce antibodies, these were really the first cellular components known to be playing a role in the adaptive immune response. Um, they knew that individuals who survived a bacterial infection were immune or at least partially immune to reinfection with the same pathogen. They would also do things like take serum from an immune patient and mix it with a fresh culture of the same bacteria. So if they had been infected with that bacteria, the cells would start clumping together in a process that was known as agglutination. So these clumping cells here are happening because you have antibodies. Now, if they weren't priorly infected, you didn't have this clumping occur. So there was no agglutination. And thus, you don't see the clumps that you do in this first one. Um, we know that this is caused by antibodies, also known as immunoglobulins. Immunoglobulin. And so we're going to talk about B cells and their role in the development of antibodies. So B cells differentiate. They are going to really be born and do most of their function in the bone marrow. So what we're going to talk about here is first related to the bone marrow. Central tolerance is one of the first things that the B cells will go through. So the B cells are going to be exposed to self antigens. So right here we have some antigens from the self. And so these are going to go through a process where it's going to basically be very similar to what was going on with T cells. If they bind to the antigens, they are going to be eliminated in a number of ways. So we could have clonal deletion. So this means that any B cell that recognizes these antigens are going to be deleted and not ultimately be producing other clones of it. Or you can also go through what is known as clonal anergy. This, they're not going to be deleted. However, they are going to no longer be able to be physically able to function. So they won't produce antibodies, but the B cell does not die for whatever reason. So if the B cells don't bind to self antigens, they are going to go down this different pathway and potentially leave the bone marrow to be exposed to other things. So here they've got through this first selection process of negative selection, making sure that they don't bind to antigens. Now you are going to go through something known as peripheral tolerance. So in the periphery, there are going to be new antigens that you're exposed to. If these antigens are bound by the B cell, that is going to be a signal for the B cell to start producing antibodies. But it's not a sufficient signal in and of itself. So we have here a receptor, or it's really a MHC protein that is presenting a antigen for the nearby T helper cells to detect. And if it's presenting something that the T helper cells uh, do not recognize, that, because they went through this whole positive and negative selection, indicates that these are indeed self antigens as well. So again, you're going to have deletion of this clone. So it will go through apoptosis and die off. If, however, the B cell does bind to the antigens and it binds to the T helper cell, then it is going to get a signal to start differentiating and reproducing. So here we actually are going to have many of these cells being produced. These are going to be your plasma cells that are secreting antibodies. Alternatively, some of these cells can become memory B cells that can lead to a stronger, faster secondary response. The subsequent challenge by the same antigen can be fairly soon in the future, or it can be many years later. Either way, you have a much expanded ability to produce more plasma cells that produce a faster overall response than you initially got with the signal. All right, so many effector cells in the immune system will have FC receptors. So the FC is the constant region of the antibody. It is formed by making two heavy chains together. So these are two heavy chains. These are the light chains. The constant region is going to be made up of the heavy chain plus the bottom part of the light chain, and then the variable region is going to be where you find the antigen binding site. So the antigen binding site is on the variable region. Because you have receptors that can recognize this means that the antibody can bind to your antigens and then stick to whatever is attached to that. So it might be an entire bacteria that's been attacked to it. Because it recognizes the FC, now you're going to have even more specificity of the effector cells that are binding to it. Now, you actually have 5 different classes of antibodies. We're going to go through each of these classes and talk about how they are similar and different from one another, starting with the IgM pentamer. So this is made up of 5 different basic antibody units. So we can see the basic unit there. It's best at fixing complement protein and is going to be the main antibody in primary responses. So this is first. What we're going to see is that the IgM is going to ultimately change to an IgA, IgG, or IgE via class switching as it progresses. But this is the primary antibody that's produced in this initial response. Thus, the antibodies made are still specific to the pathogen, but they are different in they are different antibodies, but the specificity and the antigen binding sites don't actually change when the class of antibodies change. IgG is the main antibody in secondary responses. So once the IgG antibody is secreted, it is going to be responsible for clearing pathogens and activating complement proteins. It doesn't activate complement proteins as well as IgM, but it does do a good job of clearing it. Um, it does cross the placenta to help protect the developing fetus from disease, um, but it's going to be much more important because you have this region here that's going to be recognized as something that can help clear the pathogens by binding to them, increasing opsonization, neutralizing toxins, etc. The IgA antibody, this is one that is typically going to be found in mucus, tears, saliva, and the colostrum. This is your early milk secretions after you've had a baby. This exists in two forms, so you can have a 4-chain monomer in the blood and an 8-chain structure or a dimer in the exocrine gland secretions that we're talking about up here. Dimeric IgA is the only body to leave the interior of the body to protect the surfaces. So this is going to be something that gets secreted, and that makes it unique from the other antibodies. IgE is thought to play a role in the anti-parasitic response. There's this theory or hypothesis out there known as the hygiene hypothesis that basically states that part of the reason why we develop allergies and asthma has to do with the fact that we are still producing the IgE antibodies, but because we're no longer going to be exposed to many of the parasitic things that we were when we lived in a less cleansing environment, they get targeted towards allergies, and this leads to an allergic response. This does it because it results in degranulation of mast cells and basophils. So you think histamine release, among other things, with degranulation, thus leading to allergies. And then the last one is the IgD monomer. So it is actually identical to the IgM, except that it has different heavy chains and serves as the B cell receptor. All right, so we've talked about the antibodies. Let's talk about the primary and the secondary response. So your initial exposure down here is going to lead to a primary response. This is going to be dominated by the IgM. Now you're going to clear it, you're going to go through some class switching, and now during the secondary immune response, you still have the same specificity but a different antibody targeting it. So the IgG response is going to dominate here. Now, it's not exclusive IgG and IgM, but those are going to dominate. So that the second response, you'll notice the distance from here to here is much shorter from here to here, and the height is really, in order to get to this same response, you only need to go a short distance. So this means that it's going to be faster and stronger than the primary response. Now we can take advantage of that in a number of ways. Let's talk about active versus passive immunity first, though. So active immunity is the resistance to pathogens acquired just due to the fact that you have an adaptive immune response. This can be either natural or artificial. So how can we take advantage of the secondary response? Well, we can give ourselves vaccines, which are going to provide our immune system exposure to antigens that it can then use to recognize the pathogen in the future. Whereas in naturally acquired immunity, the same thing's happening, except that it's the pathogen and that can actually cause the disease and not just the thing that's triggering the immune system. In passive immunity, the antibodies are being transferred, but you're not going to get a memory of them. So this can happen naturally during fetal development and breastfeeding when the mom delivers antibodies to the baby. And it can happen artificially with injections of immunoglobulins taken from animals that previously have been exposed to that specific pathogen. All right. So I mentioned earlier that we had T cell dependent and T cell independent antigens. Most antigens are going to be T cell dependent, but there are some that are independent. So usually T cell independent antigens are repeated carbohydrates compounds that are on bacterial cell walls. The B cell receptor has two binding sites, and this leads to cross-linking of the surface antibodies, which is sufficient to activate in the absence of the MHC2 because it cross-links there. But in T cell dependent, it's not repeated to the same degree on the pathogen, thus it's not going to have enough signals to have them cross-link. Thus, the B cell needs to receive two signals. So now you need the MHC binding to the antigen as well as the secondary CD4 binding to it. So you have the antigen on the MHC, and this leads to the secretion of the cytokines that act as a secondary response to it. So you have your activated B cell. So this is the first activation. This is the second activation. And now you have the cytokines that activate the B cell so that it starts to produce antibodies. All right, so that does it for B cells. In our next video, we'll talk about some different disorders related to the immune system.